Abiogenesis experiments confirm that first life requires intelligent design. Abiogenesis, the hypothesis that life arose naturally from non-living matter, has been an area of scientific investigation for decades. Researchers have long attempted to explain how the first self-replicating systems, the basis of biological life, could have emerged through natural processes. Despite extensive efforts, experimental results consistently show that the emergence of life is far too functionally complex to be explained by random chance or unguided processes. Rather, the data from abiogenesis experiments confirm the necessity of intelligent design, ID. Here's why. 1. Intelligent design is essential in abiogenesis experiments. Each abiogenesis experiment involves highly intricate design and control by scientists. From the creation of specialized environments to the precise selection of chemicals and reaction conditions, every step is crafted with deliberate intent. In these experiments, the scientists themselves are acting as designers, carefully crafting the conditions under which chemical reactions can take place. This human-driven design highlights a fundamental truth. To achieve even the simplest life-viable biochemical structures, intelligent intervention is required. This is exactly the point. The success of such experiments is a direct result of intelligent design by the scientists. Without careful planning, nothing resembling life forms. If the scientists did not intervene with intelligence and purpose, the processes would not result in anything close to life-functional biomolecules. This affirms the need for intelligent input in the origin of life. 2. Extreme purity requires intelligent design. Another significant aspect of abiogenesis experiments is the need for extreme chemical purity. In natural settings, molecules are far from pure. Environmental contaminants, reactive side products, and other impurities would be present. However, in these experiments, the chemicals must be of extremely high purity to even begin to form the necessary prebiotic compounds. This purity is not something nature would or could produce on its own. It takes human ingenuity and intelligent design to create and maintain this level of chemical cleanliness. Therefore, achieving the conditions for life requires a level of precision and purification that can only be produced by intelligent agents. Without this, the natural conditions on Earth would result in too many unwanted side reactions and contaminants for any life-like structures to form. 3. Intelligent design prevents gunking up in reactions. In unguided, natural chemical processes, organic reactions create a plethora of unwanted byproducts that interfere with the desired outcomes. These byproducts gunk up the system, preventing the formation of functional molecules necessary for life. Scientists, in their abiogenesis experiments, routinely employ intelligent protocols to remove these unwanted compounds, ensuring that the reactions proceed smoothly. This need for intervention highlights a key point. Without intelligent direction, natural processes lead to chaos and degradation rather than progress toward complexity. Life requires specificity and functional information, things that random chemical reactions simply cannot produce on their own. 4. Lack of complex specified functional information, CSFI. A fundamental issue with abiogenesis experiments is that none of them produce the complex specified functional information, CSFI, necessary for life. The instructions for building and sustaining life, encoded in DNA, are highly complex and precise. Yet, no experiment has ever produced the amount of CSFI required for even the simplest life form. This is a major defeater for any naturalistic origin of life scenario. Without a source for CSFI, there is no way to explain how the immense amount of information necessary for cellular functions could arise. Intelligent design provides a coherent explanation for the origin of this information, as it recognizes that complex information systems, like DNA, are the product of a designing mind. 5. Functional irreducible machinery is essential for life. The simplest forms of life require a minimum set of functional instructions and molecular machinery to survive. Studies have shown that this minimum set includes around 250 genes and their corresponding proteins. These proteins are part of complex, irreducible systems that must be fully functional from the start. Removing any one part renders the entire system non-functional. This concept of irreducible complexity poses an insurmountable defeater for unguided evolutionary processes. These systems must arise all at once, as a partially functioning set of instructions is useless for life. This necessity for fully functioning machinery from the outset points toward intelligent foresight in design. 6. Polypeptides are not proteins. It's essential to note that simply forming a chain of amino acids, a polypeptide, is not enough to create a protein. A polypeptide must have a specific sequence of amino acids that folds into a precise three-dimensional structure, forming a nanomolecular machine that performs a specific function within the cell. Without the correct sequence and folding, the polypeptide will not become a functional protein. Achieving this precise folding requires an incredibly fine-tuned process, one that cannot be achieved by random assembly. The necessity of sequence specificity once again points to the role of a designer who knows the functional requirements beforehand. 7. Protein complexes and genetic control circuits. Most cellular functions require the coordination of multiple proteins that work together in complexes, often containing between 6 and 100 proteins. These functions are controlled by genetic circuits and feedback loops that ensure the proteins act in a choreographed manner. This level of coordination cannot be achieved by chance. It demands pre-existing knowledge of how each protein and gene should function together in harmony, reinforcing the conclusion that life requires an intelligent designer. 8. Protein length and functional sequences. Bacterial proteins, on average, are 300 to 350 amino acids long. Scientific research has shown that only 1 in 10 to the power of 63 random amino acid sequences can form a functional protein as short as 92 amino acids. Given that natural selection only works on functional entities, the functional proteins must form by random chance assembly in abiogenesis. Yet, the odds of forming even one functional protein are astronomically low. 9. The impossibility of random assembly. Considering that the simplest self-replicating organism requires at least 250 proteins, and that each functional protein requires random trials on the order of 10 to the power of 63, the total number of trials needed to form the proteins necessary for the simplest life form is 10 to the power of 15,750. However, the total number of chemical reactions that could have occurred in Earth's entire history is estimated at around 10 to the power of 71, far lower than the required trials. This disparity demonstrates the empirical impossibility of life arising by random chance or natural law. The number of trials available in the history of the universe is vastly insufficient to account for the formation of even the simplest life forms. Conclusion. Naturalistic abiogenesis is not a viable explanation. The experimental evidence and data from abiogenesis research overwhelmingly confirm that life's origin cannot be explained by naturalistic processes. Instead, the complexity, specificity, and coordination required for life point directly to intelligent design. Life is not the result of random chance but the product of a purposeful, intelligent cause, one that is capable of creating the intricate machinery and information systems necessary for life to exist. Thanks for watching. Please like, share and subscribe, and comment below if you agree or would like to add your thoughts. If you would like me to respond to your comment, please make a level 1 comment directly below the video. I will respond to all polite level 1 comments even if it takes me a while, due to other commitments.